Very possible. Only correct number. Long level. अरे वो इसने का लॉक किया फिर मैंने सुना
Good job. Now this is more like it. More like the voice.
पॉकेट के अंदर रखा है
और 6 मिनट है
तुम्हारी और तुम्हारी बच्ची
अब तेरे मुंह पे होगा
ये और नहीं Will you not so much?
She's sleeping outside the balcony. That's why she sleeps. I don't know if she saw a cockroach. She saw those policemen. Yes, but when you're like
Now the next is the last law that we are going to study would be law of independent assortment. Independent assortment that we are going to study, that is a dihybrid cross. What did we study here for monohybrid cross? What was it just now? I told you what is monohybrid cross? Single bond. Independent assortment. Here, what are we going to understand is when plants with two pairs of traits are crossed. We are talking about two characters or two pairs of traits. The format will be exactly the same, only instead of one, we are going to talk about two pairs of traits. So let us see how. Here, what are the typical Mendelian traits that we have to study here? Parent plants, same like this. Parent plants, we will take round and yellow, or yellow and round. Let me take yellow and round. Or same way, you can write in any sequence, not a problem. Round and yellow seeds. Crossed with just the contrast. What is the contrast? Wrinkled and green seeds. Round and yellow with wrinkled and green seeds. This is the phenotype. We'll have to write down the genotype now. How will we write down the genotype? Round, capital R, yellow, capital Y. Wrinkled, small r, green, small y. Correct? It's exactly the same thing, only that we are now talking about two pairs of traits. Now, for two pairs of traits, meaning how is the condition if I talk about a cell from this plant? I talk about a cell from this plant. How would the pair of chromosome be? As I told you earlier also, I may understand these plants have got some 5 pairs of chromosome. So I want to see in one pair of chromosome, how is this? R and Y. It's like this. Two genes now we are studying. This is one and this is another. This gene is responsible for the shape and this gene is responsible for the color. Clear? Eh? Why are we writing twice? Because it is a pair of chromosome. Every pair of chromosome definitely has to have two alleles for a gene. अब वो दो एलील homozygous भी हो सकता है या तो heterozygous भी हो सकता है. Now we are talking about a homozygous condition. Is this clear? Remember this type of a situation, but we will not always draw chromosomes. We will always write in letters like this. So this is the the recessive homozygous, dominant homozygous, right? What are the two types of homozygous combinations possible? या तो dominant हो सकता है या तो recessive हो सकता है. Two types that we are talking about. So this is what it is, and now we have to understand gametes. Tell me how many types of gametes from these plants? How many types of gametes? What did I tell you earlier also? What are you supposed to understand? How many types of gametes? What is the situation here? It's a pair of chromosome. Capital R hai and then capital Y hai. And if these two chromosomes have to split, cell division agar hona hai, then how many types of gametes will be formed? One type. This chromosome and this chromosome. Is there any difference in the allele? Try to understand. समझने की कोशिश करो. Otherwise, if you don't know the first step itself, पूरा cross will be wrong. Absolutely सब wrong होगा. Stepwise, you should be able to understand what it is. So I'm saying, if the situation is something like this, a pair of chromosomes, how many alleles we are considering here now? Two pairs. This is one. This is another. This pair of chromosome during cell division has to split. So if it is so, and two now new cells have to be formed, gamete formation that we understand. So what the condition? It is something like this, capital R and capital Y. Are they the same type of gametes or different types? Same type. So how many type of gametes from these plants? One type. Am I clear? How many of you are still not clear about what I'm saying? How many of you still have a doubt with this? If you have a doubt with this, there's no point proceeding further. Are you clear what I'm speaking here? Capital R, capital R. Why write twice? Because a pair of chromosome, identical alleles, homozygous condition. Why write capital Y, capital Y? A pair of chromosome, identical alleles, homozygous condition. That is the reason we are writing this way. Next step, gametes, how many types? That's the next question. Gametes, how many types? Gametes, how many types? It is a homozygous condition. And homozygous, you talk about 2 pairs, 3 pairs, 5 pairs, 6 pairs. Homozygous, jab tak hai, it is always one type of a gamete because the entire chromosome will have the same alleles. So how many types of gametes? I can also write this way. This is one type of a gamete. Why do we circle to show gametes, or why do we circle to generally show that these are gametes? Earlier also did we write this way, gametes? Yes, gametes always written this way. So how many types of gametes like this? What about this side? What should we write then? How many times? One time. Homozygous है तो one type ही होना है, because the concept is this: a pair of chromosomes split like this. Is this clear? Now, next is we try understanding that F1 generation now. What about the F1 generation? F1 generation, how will we get? F1 generation, how will we get? Fertilization. Then what should the pair of chromosome be? Tell me. Then what will the pair of chromosome be? I want to write down now the pair of chromosome. What will the chromosome be now? This is one and this is another. Yes or no? ये दो chromosome, now that they come together as a process of fertilization to form the zygote, what will be the combination? The chromosome pair, will it be like this now? Yes, the chromosome pair is like this now. How will we write down the genotype? What is the meaning of genotype? Combination of alleles. So how will we write down the genotype? There are two formats of writing. I would always ask you, write this and then write this. There is no mistake that you make. Chromosome by chromosome likho. So how will I write? Capital R, capital Y, small r, small y. There is another format of writing. ऐसे भी लिख सकते हैं, where I am writing pair of allele. Pair of allele के हिसाब से भी लिख सकते हैं, या तो chromosome के हिसाब से भी लिख सकते हैं. I would ask you, follow this method. Not a problem. You can go by the other method also. There's no mistake there that way. But when you go by this method, there is no mistake. Error होने का chances कम हो जाता है. Straight away this chromosome I write, and then this chromosome I write. इसको क्या बोलेंगे? Genotype. तो इसका phenotype क्या होना है? इसका phenotype क्या होगा? Hybrid. Round and yellow. What is the law of dominance stating? The law of dominance states whenever two parents are crossed, the phenotype of the F1 generation is always resembling the dominant parent. Kabhi possible hai in the first generation recessiveness expresses? No, we had written one question also that the recessiveness will not get expressed. So what is the phenotype? We will write hybrid, round and yellow seeds. So that is the first step. Now what next? How will we get the F2 then? How can we get the F2? By self. What did we do in the previous concept? In the previous segregation mein, what did we do in the concept of segregation? How did we get the F2? By crossing F1. Yes or no? So here also we'll have to do the same by crossing F1. So these are the same genotypes. Phenotype same rahega? Exactly. What do I write as phenotype? Hybrid, round and yellow seeds. But now we have to understand that if this is the pair of chromosome, this is the condition, no, for the hybrid ones. Now if this is the condition for the hybrid ones, then now next what? We'll have to find out how many gametes from this type of a condition. Now that is a bit difficult, but you'll have to understand and try remembering that how can we talk about the type of gametes of the F1. How can we talk about the type of gametes of the F1? Now here we'll have to we have to try understanding that this pair of chromosomes, how would they behave? What probability that they show type of gametes? This is an extra concept not to include in the cross. Try understanding what I'm drawing and what I'm telling you. Capital R, small r, capital Y, small y. This is the, all of you can see, you know, the last line you can see. Now, ये मैंने क्या draw किया? This condition. F1 plant, what is the condition of the paired chromosome? Now, next, did you study a concept known as DNA copying before cell division? Yes. Since it is a heterozygous condition, we have to take that concept also now. DNA copying is a must, but homozygous में why did not we take that? Homozygous में अगर DNA copying हम consider भी करें, then also type of gametes वही same ही रहना है, nothing will change. But heterozygous definitely we'll have to take it take in consideration because as a result of DNA copying, the combinations will change. How? This is one chromosome, this is the other chromosome. Capital R hai. What is this side? Capital Y. What is this side? Small r, small y. I'm saying DNA copying is always happening before cell division. So, kaise? These are the newly formed chromosomes. This is a newly formed chromosome. This is a newly formed chromosome जो duplicated copies है. तो यहाँ पे कौन सा एलील होगा? सेम कैपिटल आर. यहाँ पे कौन सा? सेम कैपिटल वाई. यहाँ पे कौन सा? स्मॉल आर एंड यहाँ पे स्मॉल वाई. व्हाट इज इट? डुप्लीकेशन ऑफ क्रोमोसोम. डुप्लीकेशन ऑफ डीएनए मतलब एक डीएनए से दूसरा आइडेंटिकल कॉपी बनना. दैट्स इट. तो इससे अगर ये कॉपीज, हाउ मेनी क्रोमोसोम्स अभी? 1 and 2 and 3 and 4 क्रोमोसोम हो गया ना, 4 डीएनएस ना. एक से दूसरा कॉपी, एक से दूसरा कॉपी बन गया. इज दैट ऑल? नहीं, इसके बाद भी समथिंग एल्स. ये दो strands are parental strands. Nothing, no change in them. But ये जो newly formed copies है, they always have a tendency of forming a X-shaped configuration जिसे crossing over बोलते हैं. A process known as crossover, crossing over. Why are we again studying this now? Because in order to understand how many type of gametes, this is a must that you have to take into consideration again. तो ये जो दो क्रोमोसोम बना है, they will always form a X-shaped configuration ऐसा. अब alleles मुझे बताओ, if you are understanding, what is the allele here? Capital R. ये क्रोमोसोम ऐसे हुआ, so where is this allele now? This is the chromosome, no? So capital Y is here. This is the chromosome. ये है chromosome. And ये chromosome now overlapping. What about this one? Chromosome 3. ये है chromosome. What are the alleles? Small r, small y. Now that they overlap, small r, small y ऐसे हो गया. Overlap. आ रहा है समझ में? कितना तक आया समझ में? ये जो दो newly formed copies, they are just overlapping, a process known as crossing over taking place, and as a result, the newly formed copies they overlap. अब overlap होने के बाद, something more that happens is these chromosomes will break and they will इसके साथ ये join and इसके साथ ये join हो गया. Is it not? Try to understand. If you try to understand, it will go very fast. I yelling, shouting 10 times will not make a difference unless and until you try to understand. Are you able to understand the difference between these two? ये duplicated copies था. Duplicated copies में sequence of allele exactly the same. अब ये duplicated copies they have overlapped, crossing over हो गया, and now this are thread-like structures. When they cross over, turn, twist, every time there is a breakage of chromosome at this time, and then new combinations banega. Why? Because one chromosome always gets attached with the other chromosome at this time. So crossing over is the time when new combination of alleles and genes happen in chromosomes, and that's the reason we are different from our parents. We have studied variation. Do you remember variation? Why we are a bit different from our parents, even though we have got their chromosomes. नहीं तो exactly mother के जैसे, father के जैसे, we are not identical, no? So this is the reason. What is it? Crossing over is the reason. 2 minutes. Crossing over is the reason why we show variation. Now you tell me, what would be the 4 chromosome combinations now? 1 and 2 and 3 and 4. अब एलील के combinations क्या होंगे? 1 capital R, keep telling me, capital R and capital Y. What about the second chromosome? Tell me. Capital R and small y. This is a new combination. Yes or no? Tell me the third. Small r and capital Y. This is another new combination. And the last, that is the fourth, is small r and small y. So these are the four types of gametes. Now I want to write down the gametes of F2. What are the gametes of F2? One is capital R, capital Y. This is the first one. What is the second? Capital R, small y, small r, capital Y, small r, small y. Is that correct? 1, 2, 3, 4. अगर ये चार इस प्लान के गैमेट्स है, what will be the gametes of this plant? If these are the gametes of this plant, exactly the same. Whatever we studied for segregation, same tha. Now we want F2 generation. Now we want F2 generation. Now F2 generation में we'll have to make the Punnett square, and it's a big box now that we have to make. I want all of you to complete till here. Very nice, neat, you'll have to write, but this is only for understanding. इतना नहीं लिखना है. This part not to write. Write till here. All of you complete in your notebook till here. Then I will show you how to make the Punnett square. I'll be back in a minute. Don't talk. हो गया इतना? हाँ चलो अभी फटाफट make the Punnett square. All of you make the Punnett square. This is very easy, just exactly same like before. तुम दोनों का प्यार ना मुझे बिल्कुल नहीं अच्छा लग रहा है. Make the Punnett square. Method is exactly same like the previous. Already I have done for segregation. Now exactly the same. Fill in the gametes. One plant male and the other plant to consider female. And now write down the gametes. So, which gametes to now write? 1, 2, 3, 4. The same thing here you are supposed to write here. And again, when you make the Punnett square, this has to be 3 lines. If you are doing it in a ruled book, it has to be 3 lines because you'll have to write down the genotype as well as phenotype. And small r, capital Y, small r, small y. I write this side also. Don't talk, finish your work. Capital R, small y, small r, capital Y, small r, small y. So we had written down both the sides, gametes of both the parents. Now I number it 1, 2, 3, 4 so that it's easy for you to understand. 5, 6, 7, 8. How will we get? How will we get now the combinations? यही 4 gametes पहले vertically लिखना है and यही 4 gametes फिर horizontally लिखना है. And then how many columns and rows? 5 rows, 5 columns बनाना है. If it is the rule, I'll just take 10 minutes extra today. Just because this is the last topic, we are finishing up today. So this entire one single box has to be 3 lines. Remember, when you are making it in a ruled book. If it's a ruled page, remember it has to be very neat, clear. Done? Once you have done this, now start understanding the concept of fertilization. Now, can I show fertilization 1 and 2? Can I show fertilization 1 and 2 possible hai? No. 2 and 3, 3 and 4 possible hai? Why not? Gametes of same plant. तो कैसे करेंगे? 1 and 5. पहले ये ले लो, 1 ले लो, 1, 5, 1, 6, 1, 7, 1, 8. Fill in this. Okay, so let us do 1, 5. कैसे लिखेंगे? Just exactly this keep writing. Don't make any more combinations. सिर्फ एक के बाद एक लिखते जाना है. Capital R, capital Y, capital R, capital Y. Done? 1 and 6. Capital R, capital Y, capital R, small y. 1 and 7. Capital R, capital Y, small r, capital Y. 1 and 8. See if you are understanding. Doubt hai pucho. Anybody not understanding? See that you are very clear. Which fourth and this one? हाँ तो इसके साथ ये करेंगे ना. हाँ dominant always first. Always dominant जहाँ तक possible है dominant पहले लिखो. करो. Did you all understand what did we do here? करो अभी second line. 2, 5, 2, 6, 2, 7, 2, 8 ऐसे करते जाओ. 2, 5, 2, 6, 2, 7 and 2, 8. Two line, the second line is over, then go to the third. 3, 5, अब 3, 5 में देखो recessive पहले आ रहा है, try writing dominant. अगर dominant combinations है तो उसको पहले लिखेंगे, meaning 5 को पहले लिखूंगी and then I will write this. 6 पहले लिखूंगी and then I will write this. उसके बाद there's no need, I will write as it is. And 3 and 8. Done? आया समझ में? क्लियर है? एंड द लास्ट लाइन में भी मैं 5 पहले लिखूं because that is dominant. And now once this is done, understand, all of you understand and do. एक बार अगर समझ में आ गया, then any type of a combination you will be able to do this. तो पहले क्या करना है? This is a homozygous plant we have taken, homozygous recessive dominant. We have understood how to get the gametes. Then if it is a heterozygous plant but 2 pairs of alleles है, so that's the reason we have to go for this complete combination. Otherwise we would have taken only 2 gametes. नहीं 2 gametes नहीं होगा, यहाँ पे 4 gametes होगा. What are the complete combinations possible? To understand that, we have done this method, and then now you know. Actually, in simple, when you get the method, the gametes, it is actually this way: 1 and 2, and this. These are the 4 combinations that you can later understand also. But ये proper method से सीखना बहुत जरूरी है. Now let us try understanding how to write down phenotypes. Phenotypes कैसे लिखेंगे? If one capital R is there, that is enough, no, for roundness. One capital R है, one capital Y है. These are dominant traits. In the genotype you check. One capital R है, definitely it will be round. One capital Y है, definitely it will be yellow. तो wrinkled कब होगा? Something like this. When will it be wrinkled? When it is something like this. जब कैपिटल R है ही नहीं. Then when will it be green? जब कैपिटल Y है ही नहीं. Then you tell me this. Completing the box, you may do it at home as homework. I will just tell you what are the guidelines. Abhi, tell me what is the phenotype of this? Round and yellow. This is round and yellow. Tell me what will be the phenotype of this? Round and yellow. Tell me what will be the phenotype of this? Round and green. And what will be the phenotype of this? आ रहा है समझ में दक्ष? हाँ, round and green. अच्छा ये बोलो. Round and green. So when will your plant have green seeds? जब कैपिटल Y जेनोटाइप में है ही नहीं. ग्रीन एक्सप्रेस होने के लिए 2 स्मॉल y होना जरूरी है, because the recessive allele always expresses in a homozygous condition. दोनों क्रोमोसोम में होनी चाहिए. कैपिटल Y एक भी है तो येलो हो जाएगा. करेक्ट. Now tell me what will be the phenotype of this? Yellow. So this will be wrinkled and yellow. What about this one? नहीं लिखना है. इसमें ये क्या होगा? Round and yellow. Clear है? And tell me what about this? Wrinkled and green. So how many plants you are supposed to get now? You are supposed to get 4 types of plants. Which are the 4 types? One is round and yellow. How many plants you are supposed to get round yellow? 9. You are supposed to get this. Complete this at home, not a worry. Next, tell me. First you understand the method, then complete it at home, understand and do. Okay, round and yellow 9. What is the other type of a plant we got? Round and green. Round and green. How many you are supposed to get? 3. Round and green 3 होना ही चाहिए. What is the third type of a plant we got? Wrinkled and yellow. We got wrinkled and yellow. How many plants? That also you should get 3. Wrinkled and yellow 3. And last is wrinkled and green. Wrinkled and green. How many you should get? 1. Now which are the parental phenotypes? Ye and ye. These are the parental phenotypes. Yes or no? Round and yellow, wrinkled and green. तो इसको क्या बोलेंगे? These are new combinations. जैसे मैंने पिछले क्वेश्चन में भी पूछा था, recombinants. How many recombinants? How many total plants are there? 16. है कि नहीं? 16 plants. 16 plants है ना? 4, 4, 4, 4. 16 plants. Now you tell me, how many plants round and yellow? 9 out of 16. Round and green, 3 out of 16. Wrinkled yellow, 3 out of 16. Wrinkled green, 1 out of 16. Recombinants कितने हैं बताओ? Recombinants कितने हैं? 6 है. And parental combinations कितने हैं? Parental phenotypes 10 है. This and this. Clear? So phenotypic ratio of dihybrid cross. What is it? Phenotypic ratio of dihybrid cross. What is it? 9:3:3:1. Phenotypic ratio of monohybrid cross क्या था? 3:1. All of you will write down this. ये बॉक्स के नीचे ये लिख के रखो and go home and complete this Punnett square. Okay. Sex determination is the last topic which already we have completed with reproduction. We have completed, no? Sex determination I had already taken up with reproduction. Remember those chromosomes, autosomes and sex chromosomes and the gametes I have taught you along with that. Is there any doubt with that topic? So the concept of sex determination is the same. I had already taken this up with reproduction where two types of sperms, A plus X. Do you remember? A plus X, A plus Y, 23 chromosomes. So that go through that flow chart. That's the same thing as sex determination that you'll have to study. All of you will complete with this and go through. If there's any doubt, you come for the doubt solving session and try clarify. In case you are not getting this ratio, all of you should get with this and try clarifying, check if there's any doubt. Definitely come for the next doubt solving session. So with this, we end with the portion, but not a problem. There'll be again some more. If anybody needs any special extra classes, definitely you confirm it with the coordinator. There'll be some help sessions, not a problem, but provided you need, you'll have to read and come. Sit here for some time, 5 minutes, sit here.
देखिए मैं जा रहा हूँ